Thank you for joining the Once Changing the World, India's first future tech meets sustainability podcast. And today, I'm delighted and honored to have with me Dr. John Hébert, who is a professor of neuroscience and genetics at the Albert Einstein College of Medicine, author of Replacing Aging, leads the Hébert Lab. He's the founder of B Therapeutics, which aims to reverse all forms of age-related damage to the brain by progressively replacing old brain tissue with new young tissues with the intention to reverse brain aging. So, Dr. John, really appreciate you taking time being part of the podcast. I think it'd be great if you can start with a small, brief introduction, background, and also if you can explain what age reversal actually is.
Cool. Yeah. So I'll start with the background about myself for a bit. I've always been interested in fixing our fate. That as we get old, we fall apart, suffer and die. Not something that I've ever looked forward to and always wanted to change. So that drove me into science, biological science. And I did a PhD in genetics and then focused on the brain, which, is probably the most important part of who we are. And started thinking about ways that we could impact brain aging, reduce brain aging, and really realized that, aging is a very complicated process. It's a lot of what happens with age is more physical and chemical in nature than it is biological. So there's random damage that occurs to lots of components of our body, and they can't be addressed biologically as far as we know right now. And so the only solution to me seems to be to replace not just cells but whole tissue progressively over time. And I think there's a way of doing that, so removing all the damage by doing it in a stepwise fashion without losing our memories, our sense of self, and we can get into that in more detail.
Okay, perfect. So fixing fate, you mentioned, that seems to be like a huge moonshot. So when we talk about brain aging, if you manage to, when you're talking about this approach of, maybe whole tissue replacing, would that also replace a biological age?
Absolutely. So, if you put in the tissue that we're working on using as a source of replacement tissue is a very young tissue. So it's very immature. So, at most, it's the equivalent of a 1-year-old brain tissue. So it has, it should be able to last for decades. So it really is, reversing aging of the brain.
Right. So can you elaborate on this? Because obviously, the field of age reversal has seen its own ups and downs. And there are various groups, scientists, researchers who are working at the forefront, the cutting edge of aging, and they've got all of them at their own approaches. Some are looking at the senolytic treatments, some are looking at stem cells, some are looking at gene therapies, telomere lengthening. There's somebody I interviewed maybe 2 years back, was looking at blood plasma transfusion. There's epigenetic reprogramming. When you talk about this whole tissue replacing, can you elaborate on this?
Yeah, I think that's a very good question, because I think it cuts to the heart of why I think this approach is necessary to reverse aging. All the other approaches that you mentioned are centered on cells and how they malfunction over time with age. And we know that maybe the cells contribute a little bit to aging, but really what matters most is their environment that doesn't turn over time, that accumulates damage. So the experiment's been done for different tissues in the body of taking old cells from an old individual, not human, but preclinical models, and putting them in a young environment, a young body. And those cells behave now like young cells because of their environment. So the reason they're misbehaving in the old animal is because of their environment primarily. And by modifying the metabolism of the cells, it's not going to help their fate. They're still in an environment that's falling apart. So they're still going to malfunction and eventually die. So you can trick them possibly with partial reprogramming for a little while to think that they're young again, but they really won't be able to function really any longer than the cells that were there beforehand, even though the performance might get a boost for a while. The opposite is also true. So if you take young cells, so they have a young epigenome, they have young telomeres, they have young mitochondria, they have all these young features, and you put them in an old tissue, they behave like old cells, right? So the experiment in a way has been done to reverse all these things that people are working on in the longevity field. And it's been shown that it doesn't really work, or it will have a limited impact at best. So that's why I think you need not just to focus on the cells, but replacing the whole tissue to get rid of the damage that's outside of the cells as well.
So what he's saying over here is that the environment is the key. So I had read something in the similar lines, with this microbiome fecal transplant, where you take the fecal matter from healthy individual and transfer it into a sick person, and the sick person gets rejuvenated. And it's this microbiome fecal transplant has been shown to be very effective against inflammatory bowel diseases and also neurological disorders. And who might have guessed that these microbiomes, which is inside your guts, affects your cognition? And then I had spoken to Yuri Degin, who is fronting the partial reprogramming conversation for age reversal, and Dr. Harold Catcher, who's fronting the young blood plasma transfusion for age reversal. Why do you think that the young blood transfusion and partial reprogramming might not work in the long run?
Well, not going to work. It depends what your goal is. If it's to slow, stop, or reverse aging, yeah, I think it's not going to work. If it's to, get better performance or be healthier as you age, to compress morbidity so you can do better as you're aging nevertheless, then it could have benefits as shown in some of these studies, right? But in all those cases, there isn't really a significant increase in maximal lifespan. It's very negligible and hard to reproduce. There could be increases for some of these approaches in preclinical studies for average lifespan. So, I think the goal, if honestly stated in those cases, is to get more people to live, closer to the maximal lifespan of, 90, 100, and if you're a real outlier, 110 years old, right? But you're still going to be and look like, a 90-year-old, 100-year-old with a very, very limited life expectancy. So I think it just depends how you define work, So these approaches can have some impacts on health, but not necessarily aging per se or maximal lifespan.
Right. So Dr. Abid, the biologically, aging affects us the entire biology. How does age affect the brain? I mean, can you elaborate on that?
Sure. Yeah, there is a lot of variability in individuals' performance, cognitive performance over time. Some remain remarkably sharp, even into their 90s, and some start showing deficits, much earlier, 50s, 60s, 70s, 80s. So there's a fair amount of variability, even in the absence of disease. And But even the 90-year-old's brain has aged substantially. There's some very nice studies looking, functional MRI studies. Those are studies where you're looking at the activity of the brain as it's being used. And in old people that are considered to be still very sharp, they're using their brain in a very different way than young people. So young people, the functions like, different like language, face recognition, are very compartmentalized in the brain. Whereas for older people who are sharp and disease-free, they're using more and more of their brain for their routine functions. And that's because the brain is very plastic and will adapt to your needs. So those individuals, because they're using those functions, like language and stuff all the time, and they appear to be sharp, they're just using more and more of their substrate to be able to do that, because their substrate, like everybody else's, is degrading. And so they can't be as compartmentalized with those functions. So that's just one example of what happens when we age disease-free. Of course, a lot of other bad things happen with the aging brain. The incidence of stroke, cancer, neurodegeneration, lots of, if you're lucky enough to be disease-free, you still lose cognitive function, but then there's a lot of other things that can accelerate that.
Right. And along with the body, your brain, it degenerates, with age, and if we can, pause or reverse that, I think, that'll be really, really cool. Can you speak to about your approach on this, reverse aging?
Yeah. So, it sounds almost absurd at first to think that you could replace brain tissue without losing your memories, function, or your self-identity. But the clinical evidence in humans, along with lots of literature in animal models, shows that the brain, the way it encodes function and memories, is much more plastic than people generally appreciate. And the example I always like to give for the humans, even of advanced age, are studies with benign gliomas that grow. So they start from a pinpoint out and grow and destroy a part of the brain over time. And because it's over time and there's slow destruction, the functions that were encoded there get re-encoded elsewhere without the individuals even noticing that there's a problem. So for example, in the language center, a little tumor starts to grow and destroys the whole thing. Meanwhile, the individuals never lose the ability to speak. No one notices a difference in them. And at some point they get a headache or a seizure because the tumor is starting to create pressure. They go to the doctor, good news, bad news. Bad news, you have a brain tumor. Good news, it's operable. So they take it out. Now they have a big hole there. And they still can speak totally fine, even if their language center, their original language center, is gone. And so they've studied, what happens, and it moves to a new part of the cortex or the brain. And that happens with personality as well, with these tumors. And these are just examples showing how information can be seamlessly re-encoded elsewhere. As long as it's being used by the individual, it is not lost. And so that means that we can remove tissue if we do it the right way, not using tumors, but mimicking maybe how the tumors slowly destroy tissue. The other encouraging aspect of this is it seems we can add new tissue that incorporates with the adult brain. So these are, a lot of labs have been transplanting immature precursors, brain cell precursors, and showing that the neurons, the brain cells that are derived from them, integrate remarkably well with the adult brain. So this gives us a lot of hope that if we add them in the right way, that we can also not only remove old brain tissue, but add new young brain tissue. So our approach essentially is not to reinvent the wheel. So we only know one way in which our brains were made and are functional, right? That's how when they were made during, fetal and postnatal development. So that we have a fairly good understanding of how that happens. And so our approach is to mimic that to get normal brain tissue to develop in the adult using a precursor tissue that is very similar to the normal fetal human brain tissue.
So what I'm getting over here is that you will be creating these brain tissues, embryonic brain tissues to be precise. And then these brain tissues, how do you add them? Is it going to be an invasive process?
Yeah, so what we do is we analyze, very early stage of fetal brain tissue to know what are the precursor cell types that are there, what is their environment like, what signals are they exposed to, and then we recreate that, proto tissue in the lab, so that very immature precursor tissue in the lab, and that's what we've been working on and have made progress on. And then yes, it's an invasive surgery to then put that precursor tissue into the brain and watch it develop and integrate. And this is sort of the next stage of what we're doing.
So when you say that you create these tissues, how do you do that? What's the process? Is it something like these brain organoids where you take a kind of a cell and then culture it into like an organoid or a certain tissue? Is that how it works?
A little bit, yeah. So we use human iPS cells as our starting point. Those are the totipotent cells that, or at least pluripotent cells that can give rise to all the different cell types of the body. And we differentiate them into the brain precursor cell types that we need for our tissue. But instead of using the organoid approach, which is just to let them grow in these little balls, which is really not normal in structure or in cell composition, so therefore would not be functional, we try to organize them into what looks like a normal tissue. So the structure and the ratio of the cell types, the composition of the cell types is very important to get right.
Right. So once you get this tissue, where's the proof of the pudding? Before, obviously, before going to humans, we'll have to kind of show some kind of a proof of pudding. So is there some experiments that's been done on some animals or something like that, which says that this works?
Yeah. So those experiments are pretty well defined. We're just starting to do them now, but so after grafting and letting the tissue integrate and differentiate and mature, we will be looking at the structure of that mature tissue. Does it look like normal brain tissue in its structure? We will look at the wiring of that tissue with the rest of the brain. So that's connectivity, how well it receives signals and how well it projects to the rest of the brain. And then, of course, most importantly, function. So we will then test using, there's some very nice tools in neuroscience now that allows you to control the activity of neurons, either activate them or silence them using like small molecules that are given systemically to the animal. And so with these approaches, we can show that, for example, a reacquired behavior after the graft has matured is encoded in the graft, because you can silence specifically the graft neurons and show that the behavior goes away. And then once the ligand washes out that causes the silencing of the neurons, then the behavior comes back. So that pretty much demonstrates that it's the physiological activity of the neurons that you put in that are now encoding this useful behavior to the host. And this will be done in animals before it's done in humans, yes.
Right. How dangerous would be, I mean, this invasive process, and how does it merge easily? Because it's a foreign tissue, obviously. Like, how does it merge easily with the host?
Yeah, so those are two very good questions. One, is the immune compatibility? Is the tissue rejected? So ultimately, the whole field of organ transplants and tissue transplants is going towards non-immunogenic sources of those tissues, either patient-derived or autologous, so there's no rejection in that case, or some form of universal tissue that does not elicit an immune response. And, we're looking to do that eventually as well. In the short term, most efforts use what's referred to as allogeneic tissue. So that is like same species, but different individual. And that can cause an immune response. And so, people who get organ transplants, people who get cells in their brains now for Parkinson's or to treat epilepsy. There's a couple of companies that are doing this with very good results in the clinic. That's Blue Rock and Neurona, if anybody's interested. There's also another company, Aspen Neuroscience, that's going to do very well for Parkinson's. So those cell-based treatments are accompanied with a certain level of immune suppression for the individual not to reject those cells. So for now, that's still a concern unless the cells come from the patient. And Aspen Neuroscience actually is using patient-derived cells, so they will not need to use immune suppressants to treat Parkinson's. And the other question was, the surgery, is there risks for that? Yeah, there are risks of complications from any type of surgery anywhere in the body, and that's true for the brain as well. But it's not, and so, that is something we need to consider to minimize the risk of surgeries, especially if there's going to be multiple surgeries. But it's not something that precludes us from developing the approach because, neurosurgeons do brain surgeries like every day in all major hospitals around the world. And if anything, there aren't enough surgeries going on to save people that need those surgeries because of life-threatening conditions. So, right now there's a need for more surgery, and so developing better, like perhaps smart robot-assisted surgeries would be very beneficial to people who suffer from conditions already today. But then also eventually for, reversing cognitive decline with age.
When you're doing things, it sounds so very exciting, but somehow the question is why only aging? Because why not other areas of, neurodecline? I mean, because there's so many deep problems, neurological disorders, major problems, so why just aging? I mean, if you can replace this tissues, what are the other areas of, brain problems, that maybe you could sort with this?
Yeah, any type of permanent brain damage is probably going to be treated before aging, right? So the first thing we're going to use this approach for is more than likely going to be stroke damage. So permanent damage from stroke. And so that's one example. But there are a lot of other causes of permanent brain damage, trauma, tumor resections, rarely, but occasionally infections. So lots of forms of brain damage for which there is no currently treatment. And so what we're doing can be applied to all of that. And will be probably applied to all of that before it is applied to aging. But aging is just like the most exciting thing, I think.
Right.
It's like, just imagine, being sharp for a lot longer. I think is very interesting.
No, fair enough. Yeah, obviously, I mean, I believe that, if you can kind of pause or reverse aging, I think it would be like the biggest breakthrough in humankind, because, there is so much of aging population, which, stresses out the healthcare. And if we can manage to, stop reverse aging, I think it'll be really, really cool. But then there's the other side, there's millions of people in the world who are suffering with, major neurological disorders. And I see there's so much potential over there. What are the biggest obstacles you currently face in this research when you're saying that you're trying to reverse age with brain tissue replacement? What are the challenges?
I guess it's a big project. It's very ambitious, and it requires a lot of expertise and a lot of different disciplines to come together and work together to make this happen. And that, of course, takes a fair amount of funding. And so, those are probably the biggest challenges. Are to get these different groups to work together, and they'll only work together, if you have funding for them to work together. So yeah, I think that's probably the biggest challenge.
Right. Where do you see the field heading in another 5-10 years?
So I mentioned at the outset that I'm transitioning. So I'm actually leaving my position at the university and leaving my position in the company that I founded to start government programs aimed at achieving these goals. So, that's the plan I have, is to start programs that have a lot of resources to, a lot more resources than you can get otherwise in academia or even the startup industry for biotech. And hopefully that will accelerate and allow me to bring people together to work on this problem and reach a goal that I proposed to the government, this government entity, which is ARPA-H. I proposed to them in 5 years to have a proof of concept in a preclinical study that the replacement tissue works, meaning that it can encode useful information to the host. They're actually even more ambitious. They said they pushed me to propose to be in the clinic in 5 years. So we'll see. Definitely going to try and do that. But I think it's very ambitious, but not impossible. So.
Congratulations, and wish you the very best on that. Would you be able to give us a brief synopsis on your book, Replacing Aging?
Sure. I mean, I'll start off by saying that that book was written by me in 2018, so already, a good 6 years ago. So although the general concepts are still accurate, it's I would say it's a little outdated, and I would love to have the time to write an updated version, but I don't have the time right now. But the general ideas are the same, and the book is divided into 3 parts. One is just sort of giving an introduction to what we know about the process of aging that occurs in living creatures, including humans, and why a lot of the approaches that are favored today, or at least focused on today, to tackle aging are going to fail. So that's sort of the first part of the book, and we talked about this a little bit earlier on. The second part of the book is why replacements, are going to work. And they're already used in regenerative medicine to, save people's lives that have organ failure or organ damage from trauma, for example, or any other part of the body. Every part of the body has been surgically replaced except the brain. So that approach is not that novel, but it's not being applied to aging, but it could be. So that's the second part of the book. The third part is focused on the brain, and that's sort of what we've been talking about, how you could reverse brain aging with the same concept of replacement to get rid of all the damage. So that's the book in a nutshell.
I had interviewed Dr. Sergio Canavero. He was, I think, one of the first guys in the world who was thinking about a full head transplant. Obviously, I mean, because of ethical concerns and issues, the project never really saw the light of the day. Do you even think that something like that is possible, where, you would be kind of replacing body parts? I mean, maybe, because we already do it, today. I mean, for prosthetics, we do, so your thoughts on that?
Yes, I mean, it's never been done in humans, but Sergio Canavero was not the first person to think of this. I mean, there's a Russian group that did head transplants in the 50s, I think. And although they were not replacing heads, they were adding a head to an animal. But in the 70s, there's another guy, Robert White, who did actual head replacements, or body replacement makes more sense than the head replacement. Body replacement using monkeys. And, that was highly controversial, but he did have some success. So the heads were alive for a while and responsive and alert and conscious. So that's been to some extent done in terms of a proof of concept. So it's not impossible. And this guy, Robert White, was an advisor to the Pope. I mean, he was very moral and thought that we needed to do this to save people's lives that had, degenerative wasting conditions. And he happened to be a good surgeon. So, this is an idea that's been out there, and although controversial, does have its supporters.
Right. So you think that in the near future, we could have like a full head transplant?
I mean, technically, there's no reason not, right? The challenge there is the spinal cord necessarily needs to be cut, and how well it can reconnect biologically is still an open question. So it's not clear how well that can happen. But already we see, a lot of progress that can circumvent the need for doing that, although that would still be the best way of doing it is biologically. But you could circumvent it with brain-machine interfaces, for example. So there's several companies, BlackRock, Neuralink. I always forget the name of the third one, but there are probably more than three. In fact, there are more than three. And so, they're developing these in large part to re-give paralyzed individuals or quadriplegic individuals control of their body again, without reestablishing a biological connection. So it's just bypassing the brake in the spinal cord. So the surgery itself, I think can be done, and then the spinal cord issue can be circumvented, or we'll find a way of actually biologically repairing it. So, so I don't think technically that's a big challenge compared to like what we're trying to do with the brain, for example, which is much harder.
Right. So as a neuroscientist, how much do we actually know about our brain? Because 80 billion neurons, 100 trillion synapses, the way they fire and wire gives out all 5 senses, but still we know very, very little of the brain. I'm personally vested into metaverse, and in virtual reality, we have this like holy grail of the virtual reality is like, like a full dive VR, where, someday you'll be able to kind of create these virtual worlds, which will be indistinguishable from reality. So the idea over there is like, if you are able to completely structurally and functionally scan the entire cognitive structure of a human brain, then you don't really need to maybe look at age reversing. You could like upload it on a digital substrate, and then you could tweak it to so maybe I've just gone a little way off. But the question is, how much do we actually know of the brain? Are we in the position that maybe someday far off in the future, you think that we'd be able to completely structurally and functionally scan the brain, human brain?
Possibly, but it's sort of a more of a existential philosophical question at that point, because if you could do that, you would be making a copy, and are you happy with a copy of you? Or if you can make one copy, you can make 100 copies of you, right? So there could be 100 uploaded minds of you, and then there's still the biological you. And are you happy with having copies of you while you still, degrade over time, get old, and lose your mind? Because those copies will be fine without you, but you're not going to be fine with them. So if you destroyed all the copies, for example, it wouldn't affect you, right? You'd still be as conscious and as still be aging. So it depends whether you think having a copy of you is good enough for you. Like, you don't care if you yourself actually succumbs to aging and cognitive decline. I've talked to people who see it both ways, but some people prefer that they continue, not a copy of them.
Don't you see that maybe somewhere down the lines, when you're doing like a whole brain tissue replacement, isn't there over there also maybe you're starting with adding a certain new tissue onto the brain, and then eventually, obviously, if you want the complete regeneration, wouldn't that mean that right now you're just adding a bunch of tissues? Eventually, maybe you might want more changes. You might keep on adding more. So does that still retain your identity, because you're adding something completely new anyway? Though it's you're not completely changing the entire cognitive structure, but eventually, if the technology permits you to do that, because you would want more augmentation, because I guess we are forever seduced. We all say that, oh, I don't want big breast or big this or whatever, but then we are completely seduced all the time. And if the opportunity presents that, oh, okay, you got this, now you want more, because we humans are greedy. So wouldn't that mean that we would progressively
augmentation, I guess, is possible in theory. It's not something we're just thinking of maintenance, not augmentation. But I guess in theory it is possible. It's just we don't really understand how we would augment, because, like you say, there's so much we don't understand about how the brain works. And to augment function, you probably would need a much better understanding. So it's not something that at the time now we have a clue as to how we would go about doing, but in theory it should be possible. But the best I think we're hoping for here is a continuity of self, right? So if we do the replacement in a way that allows the normal plasticity of this tissue to re-encode information, memories, function, self-identity in a seamless way from one part of the old substrate to the new substrate, I think that's the best we can hope for.
Right. Right. You mentioned that the biggest challenges to the approach that you have is funding. What are the other challenges? And do you see, maybe artificial intelligence somewhere possibly helping in your research field?
Yeah, I think the artificial intelligence is starting to help a little bit. It's not much of what we do is experimentation and adjustments, and there's the current artificial intelligence can't really contribute to that, right? Because artificial intelligence, as it stands now, requires large data sets. And we don't have large data sets to feed into a learning model. And so for that, it's not terribly useful. But for a lot of things like automating very tedious tasks like growing stem cells, they really require right now a human scientist to babysit them and watch the progression of these cells. And, AI combined with, the robotic and imaging capacity can potentially do a much better job at that. And so there's a lot of examples like that where a certain level of automation driven by smart robots would be very useful in moving the field forward. And there are groups developing these AI-driven tools. So hopefully they will start to be impossible near future.
What are the next steps for your lab to bring this research closer to clinical trials? And would the transition that you mentioned about ARPA-H, what role is that going to?
There is
work of yours?
Well, hopefully it'll play a big role. They typically, the budgets for these programs is quite large. And that should help a lot bring people together to work on this. And so, there's still a lot of steps along the way that need to be achieved. So one is, the source of the tissue. What the approach we're taking is to sort of reverse engineer normal fetal-like human brain tissue. But there are potentially other approaches that, we can explore with the resources that we'll have. And then it's just a matter of testing these after grafting in preclinical models to see how well they can perform before doing it in humans.
Besides your approach, are there like any other approach you think? I mean, anybody else who's working on age reversal, not just tissue regeneration? If there's any other approach you think could lead towards age reversal?
Yeah, I don't think so, and certainly not the ones that are being actively pursued now, like some of the ones you mentioned early on, like senolytics, partial reprogramming, plasma dilution or plasma transfer. Again, those could have some positive impacts, but they're not tackling aging per se. And yeah, I don't see them working, no.
And what would be the timelines that you would give for your approach to become clinically ready?
So for the brain, yeah, again, very ambitiously 5 years. I think we can potentially achieve, clinical implementation for something like stroke in 5 years. That would be the goal. It's still ambitious, but doable.
Right.
So,
right.
And that will start, if we have that going, then I think there'll be a lot more interest and we can move a lot faster towards neurodegeneration, cognitive decline.
Perfect. We wish you the very best. Can you leave us behind with your vision of what the world is going to look like maybe in the next 10 years when all of these techs kind of converge and there's these real world utility comes out, Can you paint a picture of what the world is going to look like in the next 10 years?
Yeah, that's very challenging given that, in an ideal world where we're allowed to pursue science and technology without the constraints of changing governments and social structures, then I think 10 years would look super bright. But given the uncertainty in world politics and current governments, you never know what can happen. and so I wouldn't venture to guess. I mean, there are influences here that are sort of beyond the scope of science and technology. Fingers crossed that we can keep doing what we're doing.
Right. Thank you, Doctor. Really appreciate you taking time being part of the podcast. I think we're living in a fantastic point of time, but yes, we also live in a uncertain time, and I hope that scientists, researchers such as yourself, I mean, I think these things might look uh, very freaky right now to us because it's beyond the norm. But I'm sure maybe 20 or 30 years down the line, we'll be saying, or kids will be saying, why the hell did you guys didn't do that earlier? So yeah, I hope that these technologies is available for everyone. And I hope that we find a way, whatever approach. I don't really know what approach could lead us to, brain, age reversal, but whatever approach gets there, I think we the common people will be really, really happy. So, I mean, kudos to researchers, scientists such as yourself who are forefronting this technology. Wish you the very best. And to my listeners, if you like what you see any other, please press the subscribe button. Until next time, see you there. Thank you. Thank you, Doctor.
Thanks.
Appreciate this. Yeah.
Thank you.
Thank you. Thank you.